Hello everyone and welcome to Class 11 Biology. Today we'll discuss the second part of the Chapter 9 Biomolecule, that is Biomacromolecules or Macromolecules. So the topics are protein, polysaccharides, nucleic acid, and nature of bond linking monomers in a polymer. So let us first recapitulate the previous video. That is, we discussed that all the chemical components, molecules present in the living organism are termed as biomolecules, and these biomolecules are classified based on its molecular weight into two categories, that is micromolecules and macromolecules. Micromolecules, some examples are amino acids, sugar, lipids, and nucleotides. And there are three major macromolecules, and those are protein, polysaccharides, and nucleic acid. So we'll start with protein. Proteins are polypeptide. These are composed of number of amino acids which are linked by a bond called as peptide bond. And in this protein, it is composed of 20 different types of amino acids. That is why the protein are called as heteropolymers, because different amino acids make up a protein. It is not the same type of protein, same type of amino acids. So in this picture, you can see that these are the different amino acids. Amino acid number 1, 2, 3, 4, 5, and so on. And you can find different combination, different amino acid combination forming a polypeptide chain. Okay, so this polymer made up of monomer. Each amino acid here is a monomer, and this complete chain of amino acid is a polymer. So the polymer of this amino acid forms a protein, and in this polymer the bond is peptide bond. Okay, and each monomer here, each unit here is different. They are not same, completely same, so they are different, so it's called as heteropolymer. And the component, that is amino acid, it can be essential or it can be non-essential. So essential amino acids are those amino acids which has to be procured, or we can say which has to be obtained through a diet, because these essential amino acids cannot be synthesized or cannot be made by our body. Okay, and then there are non-essential amino acids which are synthesized by our body through metabolism, so they are called as non-essential amino acids, so they need not be taken through the diet necessarily. And then there is a third type, that is semi-essential amino acids. So semi-essential amino acids are those, again, amino acids which are synthesized by our body but not in enough quantity. So they may be required more during a particular stage of a life. Okay, so these are the three types of amino acids forming a protein. And in this protein, we can see that it starts with the first amino acid, ends with the last amino acid. Here, the example, in this example, you find 25 amino acids here, and here, that is, we have two end, here this end, and then we have the one at the 25th amino acid. So here, this end, we call it as N-terminal, because in the amino acid towards the left side there is an amino group here, so N-terminal, and towards the right side we can find carboxyl group here, this carboxyl group. Okay, so each one has its own carboxyl group, so the last amino acid has a carboxyl group here. So this is a C-terminal and this is N-terminal. Now let's look at different protein and the functions. So first one is collagen. Collagen is a very important protein, and it's one of the most abundant protein in the animals. So the function is they are intercellular ground substances. Okay, in the tissue and also, in the tissue. Next is trypsin. So trypsins are an enzyme which helps in digestion of protein. Okay, so this trypsin helps increases the rate of reaction. So enzymes are the biocatalysts. So trypsin acts as an enzyme which helps in the breakdown of protein, that is digestion of protein. Next is insulin. Insulin acts as a hormone, which again helps in regulating the glucose, blood glucose in the body. Okay, deficiency of this insulin will lead to diabetes, that is hyperglycemia. Next, antibody. So whenever we have any infection in our body has our own defense mechanism. Okay, so our body try to fight off the pathogens or the intruders or the infectious agent by producing a protein called as antibody. Next, a receptor. So these receptors are a special protein present on the sensory receptor cells, okay, which receives the signal, which receives or detects the, or you can say which receives the signal, or you can say stimuli. Next is GLUT4. GLUT4, it enables the glucose transport into the cell so that the cell can break down the glucose to obtain energy, or it can be built up. It can be used to build up and store the glucose in the form of glycogen. So these are some of the protein and their functions. So now let us look at the structure. Protein structure, the first one we can talk is primary structure of protein. So the primary structure of protein is a very simplest level. It's the first level of protein structure. It's very simple. It just deals with the sequence of amino acid in a polypeptide chain, or you can say it just deals with the, let's say, the order of amino acid. What is first amino acid, what is second amino acid, and so on. Okay, so this is a simple structure of a primary structure of the protein. So again here you can see that it starts with the N terminal and ends at the C terminal here. So next is secondary structure. So secondary structure is formed by the folding of the polypeptide chain due to the interaction between the atom of the backbone. Okay, so these are two types, alpha helix and beta pleated sheet. So here let us look at this. So in alpha helix, okay, it forms a curled, or you can say helical structure, like a curled ribbon. In this case, what happens is that the carbonyl of one amino acid is hydrogen bonded to amino H of the amino acid that is 4 down the chain. So here you can look at, just look at here. The carbonyl group here, carbonyl CO, it is bonded with carbonyl group. Okay, so this is one amino acid here having, you can see, NH, okay, amino group, and then C, R, that is a side chain, and then carboxyl group here. So we have a CO. So this carbonyl group, in this case, this carbonyl group is bonded with the amino group of the other amino acid through hydrogen bond. And because of this pattern of bonding, it pulls the polypeptide chain into a helical structure. So this alpha helix has a helical structure, and in the case of this, there are two types of bond formed. First one is peptide bond. As we have discussed, peptide bond is a bond formed between two amino acids. Okay, so the first amino acid and second amino acid, they are linked by peptide bond, and second and third is also linked by peptide bond, thus forming a polypeptide chain. So in alpha helix, there is a peptide bond as well as hydrogen bond. Okay, so this hydrogen bond, what it does is it binds, or you can say it held together the H of one amino group and then carbonyl group of the another amino group, or other amino acid. So this is one amino acid, this is next amino acid here. Okay, if you count, give a number, so this is one of amino acid number 1, 2, 3, and then this is fourth amino acid, and so on. Okay, so the bond form is between the first amino acid and the fourth amino acid. Okay, there is a hydrogen bond. Now we have another type of secondary structure, that is beta pleated sheet. So in this case, we have two or more segments of polypeptide chain like this here. This is one polypeptide chain, this is another polypeptide chain, which are lined next to each other. Okay, so they form a sheet-like structure, and these are again bound by these two strands, or two segments of the polypeptide chain is bonded by hydrogen bond here, hydrogen bond. So again here in this case you find peptide bond as well as hydrogen bond. Now the third level is tertiary structure. So tertiary structure, it gives us an oral 3D structure of polypeptide, and here we can find an interaction between the side chain of the amino acid. So for example, here we can see that this is a chain of polypeptide. Okay, so chain of amino acid forming a polypeptide here. So here in the chain of polypeptide, you can find peptide bond. You'll also find hydrogen bond, and other than that, the R group here, they will also form a bond, that is ionic bond. This side chain here, the side chain will form an ionic bond here, and then again here there are some amino acid which has sulfur in them, and these sulfur again form a bond called as disulfide bridge or disulfide bond. Okay, so this is just the chain of amino acid, that is polypeptide chain. They folded upon itself, forming a 3D structure which becomes a functional protein. Okay, so in this case, other than peptide chain, you find hydrogen bond, and then there is also a ionic bond. There is also a disulfide bridge or disulfide bond. Now the last one is tertiary structure. So tertiary structure is nothing but, again, it's a 3D structure where you have more than two individual polypeptide chain. So if there are more than let us say two or more than two subunit. So if there are two or more polypeptide chain, then they get arranged. They are arranged in such a way that they function as a single functional unit. Okay, and such an arrangement give us such an arrangement of this subunit give us a 3D structure. That 3D structure is called as quaternary structure. So example here you find is hemoglobin. As you know that hemoglobin, it helps in carrying oxygen through our body circulatory system, and this hemoglobin is present in RBC. Okay, that is red blood cell. So this hemoglobin is made up of 4 polypeptide chain. It is made up of 4 polypeptide chain. That means there are 4 subunits. Okay, 2 alpha chain and 2 beta chain. 2 alpha chain and 2 beta chain. And the arrangement, okay, give us a structure, and that structure is called as quaternary structure. Okay, now let us look at another biomolecule, that is carbohydrates. Okay, so these carbohydrates are often referred to as saccharides, and these are classified into monosaccharide, disaccharide, oligosaccharide, and polysaccharide. Okay, so monosaccharides and disaccharides, and then these oligosaccharides. These three have a lower molecular weight. That is why they are placed under micromolecules. So here, polysaccharides are the type of carbohydrate where the molecular weight is higher than 10,000 Dalton. So this is an important macromolecule in the living organism. Next. Okay, so let us just look at the classification of carbohydrates. Okay, we have monosaccharide, disaccharide, oligosaccharide, and polysaccharide. So monosaccharide, this, okay, have only one sugar molecule. This has only one sugar molecule. You cannot break it further into a sugar unit. So examples are glucose, fructose, and galactose. Disaccharide, okay, on hydrolysis it will give 2 sugar, so called as disaccharides. The examples are sucrose, lactose, and maltose. Oligosaccharides are the type of carbohydrates when it is hydrolyzed, when it is broken down, it gives us a sugar unit from 3 to 10. And then those carbohydrates are, okay, it can be like raffinose or it can be stachyose. Now the last type of carbohydrate, which is macromolecule, is the polysaccharide. So it will give us 10 or more sugar molecule upon hydrolysis. So let us talk about all these one by one, and then we'll focus mainly on this polysaccharide because this is a macromolecule. and this polysaccharide is composed of this unit called as monosaccharide. Okay, so let us say first one, monosaccharides. So these are the simplest group of carbohydrate and they are often referred to as simple sugar. So let us just look at the general formula. The general formula is CnH2O. So these sugar are made up of carbon, hydrogen and oxygen. Okay, and the ratio is 1 carbon is to 2 hydrogen is to 1 oxygen. So the ratio here, if you just look at here, the ratio of the H2O is like 2:1. Now, these monosaccharides are further classified on the basis of number of carbon. So, okay, that is N. I have written N can be 3, N can be 4, N can be 5, N can be 6. Okay, so they are further classified. And then classification can also be based on, so there are a classification of this monosaccharide. Okay, the criteria differs. First one is the number of carbon, and second classification is the functional group present in that, whether it is aldose or ketose. Okay, so let's look here. Monosaccharide. Okay, so based on the number of carbon, so if it has 3 carbon, it's become triose. The examples are glycosidic, and if it is 4, we call it as tetrose, erythrose. If the number of carbon is 5, pentose. Example is ribose. And then if the number of carbon is 6, hexose. Example glucose. And if the number of carbon is 7, monosaccharide is heptose. Example is glucoheptose. Okay, so this is how the monosaccharides are classified based on number of carbon. Monosaccharides are also classified based on the functional group. So this, based on the functional group, it can be aldose or it can be ketose. So first one, let us look at the aldose here. We can see we have aldehyde group CHO here, CHO here. So these are the aldoses. So example, glycosidic, ribose, glucose and galactose. Now ketoses have a ketone group CO, and this CO, unlike this, okay, it is not terminal here. In the case of aldoses, the aldehyde group is in the terminal, okay, at the site we can see, but in the here the ketone is not at the terminal, it is subterminal here, C double bond O, ketone. Okay, so the monosaccharides are classified first based on the number of carbon. Okay, so based on the number of carbon, if the carbon is number of carbon is 3, this becomes triose sugar. If the number of carbon is 5, pentose. If the number is 4, tetrose, and if it is 6, hexose, and so on. Okay, so depending upon the number of carbon here. So here 1, 2, 3. So this is a triose sugar, and here 1, 2, 3, 4, 5. This is a pentose sugar, and then 1, 2, 3, 4, 5, 6. So there are 6 sugar, carbon, so it is a hexose sugar. So this is how the monosaccharides are classified based on number of carbon. Another way to classify the monosaccharide is, as discussed, based on the functional group. So based on the functional group here, we have a CHO group, that is aldehyde group. So all of them have the aldehyde group. So these are the aldoses. And if it has a ketone group here, these are the ketoses. So the ketoses example are dihydroxyacetone, ribulose, and fructose. Okay. Now this monosaccharide can be written like this, long chain, or it can form a ring structure like this. Okay, so it can be long chain structure or it can be ring structure, but the number of carbon will be same. Here the example here again, 1 carbon, 2, 3, 4, 5, 6. This is hexose. Okay, here this is oxygen, right? So here 1, 2, 3, 4, 5, 6. Okay, so this is a ring structure. Let us look at other ring structure of monosaccharides. This is glucose. Glucose is a hexose having 6 carbon atom. So here 1, 2, 3, 4, 5, 6. Glucose. And then let's look at fructose here. 1, 2, 3, 4, 5, 6. Okay, this is hexose. Okay, here it's written hexose. And then talking about galactose. 1, 2, 3, 4, 5, 6. Now let's look at pentose. 5 carbon. 1, 2, 3, 4, and 5. Okay, so deoxyribose and ribose. So these are some of the monosaccharides, and these monosaccharide have a aldehyde, free aldehyde group and free ketone group. That is why they are the reducing sugar. They are called as reducing sugar. Okay, so reducing sugar are the sugar having free aldehyde or ketone group. Okay, and they can reduce cupric ion of Benedict test. So this you can find when you do a food analysis, food test. Okay, so if you want to find out the presence of glucose or the presence of sucrose and so on, starch and so on. Okay, so especially here when we talk about the presence of glucose, okay, we do a Benedict test. So if it has reducing sugar, then what they do is they can reduce cupric ion into cuprous ion. Okay, and non-reducing are those sugar which do not have free aldehyde or ketone group. Now next is disaccharide. Disaccharides are again a type of carbohydrate. When they are hydrolyzed, when they are broken down, it gives us 2 unit or 2 monomer of sugar. That is why they are called as disaccharides. So the example here is sucrose here. So the sucrose is made up of this glucose and then fructose here. Glucose and fructose combined to give us sucrose. This sucrose, when you break down, we get 2 monomers, 2 monosaccharides, that is glucose and fructose. So this sucrose is a disaccharide. Another example is lactose. So lactose, when we break down, okay, when it is hydrolyzed, it gives us galactose and glucose. Okay, this is disaccharide. Another example is maltose, where it is composed of 2 glucose, sugar. So when this maltose is broken down or hydrolyzed, we get 2 molecules of sugar, glucose. Next is oligosaccharide. This oligosaccharide, when again is a type of carbohydrate, when hydrolyzed, give us from 3 to 10 monosaccharide unit. Okay, and this oligosaccharide are classified into trisaccharide, tetrasaccharide, penta, hexa, and so on. Okay, so let us look at one such example. So the example of oligosaccharide, that is raffinose here. Okay, raffinose, when it is hydrolyzed, it gives us 3 unit or 3 different sugars, that is galactose, glucose, and fructose, because this raffinose is made up of these 3 sugar unit. Next, polysaccharide. So polysaccharide is one of the important macromolecule in living organism. So when these polysaccharides are hydrolyzed, then it yields more than 10 molecules of monosaccharide. And they are usually tasteless. So they are non-sugar, and the general formula is C6H10O5, okay, times and N times. Now let us just look at the brief classification of polysaccharides. Polysaccharides are classified based on the monomers present. Okay, if all the monomers present in the polysaccharide are of same type, we call it as homosaccharides, so homopolysaccharides. And if it is of different types, then heteropolysaccharides. So homopolysaccharides and heteropolysaccharides. So these are the 2 types of polysaccharide depending upon the type of monomers present in them. So this homopolysaccharide, it can form a storage or it can form a structural part of the body So storage examples are starch and glycogen, okay, in plants and animal respectively. And then structural, that is plant. In the case of plant, there is a cellulose, and in the case of animals, okay, that is a chitin, which forms an exoskeletons of certain arthropods. Now, talking about the hetero polysaccharides, these are composed of like different types of monosaccharides. Monosaccharides here. So the examples are heparin, chondroitin, sulfate, hyaluronic acid, and keratin sulfate. So these are some examples of polysaccharides. Okay, starch, as you know, these are the storage carbohydrate in plants. Glycogen are again storage carbohydrate in animals. And then cellulose, it forms a structural part of a plant, okay, where this, you can mostly see is in the cell wall of a plant. Now starch, okay, is as discussed, it is a homopolysaccharide. It is made up of a repeating unit of glucose, a polymer of glucose. So these starch have again like amylopectin and amylose. So amylose here, okay, we have these different types of glucose here, which are arranged one after another here, forming a polysaccharide. And then amylopectin, it forms a branch structure here. Okay, so each sugar is bonded with other sugar by glycosidic bond. So let us say this is one glucose, this is another glucose, okay, and number of them forming a polymer of glucose. So the bond between one glucose to the next is called as glycosidic bond, and the bond formation we shall discuss in another topic. So in the case of starch, since there is a pocket formed here, a pocket formed like this, okay, the iodine, when you test with iodine, it forms a dark blue color. It's all because of the structure of this, or you can say the arrangement, or you can say orientation of this glucose. Now next is cellulose. Cellulose is again a polymer of glucose. Then how is it different from starch? In the case of starch, it has two structure. Okay, so we have a helical structure straight, and then you have a branch part. But in the case of cellulose, it's a straight. It's unbranched. It is made up of number of glucose. Again here, this is one glucose and another glucose, and the bond formation between them is called glycosidic bond. So this cellulose is found in plant, which provide the rigidity to the plant, providing support to the plant. Next, glycogen. Glycogen is again a polymer of glucose. And in the case of animals, this glycogen acts as a storage. Okay, so the excess of glucose, are converted into glycogen, and it is stored. So here you can see that these are, okay, 6 hexose. These are the glucose, and we have a chain of glucose, right? And forming a polysaccharide, and this chain is a branched. It's a branch structure. Glycogen have a branch structure. Now next is inulin. Inulin is the polymer of fructose. So fructose, okay, carbon 1, 2, 3, 4, 5, and 6. So number of fructose again linked by glycosidic bond. It forms inulin. Now we have chitin. It's a polymer of glucosamine. So this chitin has an acetyl group. Okay, it has an acetyl group, and this chitin forms a tough structure of the exoskeleton of insects and other arthropods. Okay, so this is about the polysaccharides. Now there are other polysaccharides where other than sugar, other chemical components are present, and these are hetero polysaccharides. Now let's talk about the last part of the macromolecule, that is nucleic acid. So nucleic acids are the polymer of nucleotide. polymer of nucleotide or polynucleotide. So it is made up of a chain of nucleotide, and it's of two types, DNA and RNA. So let us just briefly talk about what is nucleotide. So nucleotide is a chemical component composed of three main components, that is nitrogenous base. Nitrogenous base is a heterocyclic compound rich in nitrogen, nitrogenous base, and then sugar, pentose sugar, sugar consisting of 5 carbon, pentose sugar, and then phosphate group. So all these three components together makes a nucleotide. Okay, so here this is phosphate, this is sugar, and this is one of the nitrogen base. So from here, this is one nucleotide, and this one nucleotide bonded with next nucleotide, and so on, forming a chain of nucleotide. Okay, chain of nucleotide, we call it as polynucleotide, and together we call it as nucleic acid. Okay, so nucleic acid is a polymer of nucleotide. So it is in two forms, that is DNA and RNA. So let us talk about them one by one. So first one is DNA. DNA is an important genetic material in all the living organisms. Okay, in some of the viruses, they also have RNA as a genetic material. So DNA, deoxyribonucleic acid. In our body, it carries the information. It carries the genetic information, and which is expressed in the form of proteins. And this DNA structure, the most stable form of DNA structure is B form, bDNA. So let us talk about its structure. Okay, so it is a double helix structure. So we have one strand of DNA, one strand of polymer of nucleotide here, you can see, and another strand here. Okay, so these are the double-stranded structure. These are the double-stranded structure which are twisted like in a helix. So this is a double helical structure, and this double helical structure of DNA was proposed by Watson and Crick. And the orientation. So here you will find there is 5 here, and then you can see 3 here, and 3 here, and 5 here. So 5 prime, 3 prime, 3 prime, 5 prime. So this one strand, you can see that in this one strand of DNA, the direction is 5 prime and 3 prime here. But in this strand, the direction is 3 to 5. So the direction is opposite, but the orientation is parallel. So such a DNA, we call this anti-parallel orientation. Anti-parallel. Okay, they are parallel, but the direction is opposite. 5 prime to 3 prime here, it's running from 5 prime to 3 prime here, but here it is from 5 here to 3 here. So they are opposite in direction. So anti-parallel. And in the case of DNA, here it looks like a ladder. It looks like a ladder which is twisted like a helical structure. So in this ladder, we can see that these are the backbone here. This is a ribose sugar, phosphate, ribose, and so on. So they form the backbone of the DNA. So nitrogen base, that is ATGC, they extend towards the inner part of it. Okay, so the nitrogen base extend into the interior in pairs. Okay, so here this is towards the pair, this is towards the pair, and then the one nitrogen base of a strand of DNA pair up with the opposite strand, by hydrogen bond. So they are bound to each other by hydrogen bond, and the bonding is very specific. A always pairs with T. Okay, this is nitrogen base. Okay, nitrogen base, sugar and phosphate together forming a nucleotide. Here, these are the nitrogen base which are projected inward. And A always paired with T, G always paired with C by hydrogen bond. Again, you can look at the number of hydrogen. Between A and T, there are only 2 hydrogen bonds. Between G and C, there are 3 hydrogen bonds. Okay, so this is the structure of DNA. And then again here, if you know the sequence of this nucleotide on one strand of DNA, you can easily predict the sequence of the sequence of this nucleotide on its opposite or on its complementary pair, because the pairing is always specific. They are specific. A pairs with T, G pairs with C. Okay, so another one is this. Since we discussed that DNA is a double-stranded helically coiled structure, helically coiled. So when they are coiled, if they turn 360-degree angle, that is one complete turn. So let us say here, from here to here, it is a 360-degree angle. Okay, so in this 360-degree angle, the number of base pair, that is nucleotide, is 10. Okay, so from here to here, let us say 1, 2, 3, 4, like this you can count. So there should be 10 base pair. And the distance or the position it occupies is, or the space it occupies is 3.4 nanometer. 3.4 nanometer. Okay, so in each turn, there are, one turn, there are 10 base pair. One turn is also called as pitch. So 10 base pair, and this 10 base pair occupies a space of 3.4 nanometer. 3.4 nanometer, and the width, width is 2 nanometer throughout the length of DNA, because these are parallel. Okay, the width is 2 nanometer. From here to here, it is 2 nanometer throughout the length of DNA. So this is about the DNA, which act as a genetic material. If there is any change in the structure or any change in the sequence of DNA, of course the expression of this DNA also will change. Now next is RNA. RNA, ribonucleic acid. How is it different from DNA? Okay, in the case of RNA, it is a single-stranded structure. It's a single-stranded structure. Okay, it's a polymer of nucleotide. Okay, so it will have ribose sugar and one of the 4 nitrogen base. So here, in the case of RNA, instead of T in DNA, there is uracil here. Okay, and then ribose, nitrogen base, and phosphate group, same as that of the DNA, but instead of here U in the case of DNA, there is T, and here it is single-stranded, but in the case of DNA it is double-stranded. And another important sugar is ribose here. In the case of RNA there is a ribose sugar, but in the case of DNA there is deoxyribose sugar. Okay, now these RNA, they function in various forms. Okay, so there are 3 different types of RNA. That is messenger RNA, which carries the information from the DNA. Okay, and then ribosomal RNA, it acts as a structural RNA, and then transfer RNA. Okay, so these are the 3 major types of RNA. But in some of the viruses, this RNA also acts as genetic material. Okay, such a virus, we call it as retrovirus. For example, HIV. Okay, so that's all about the biomolecules, or you can say macromolecules. So in the living organism, there are it is considered of course there are 3 macromolecules or 3 macrobiomolecules or biomacromolecules. Those are protein, a polysaccharide, and nucleic acid. And sometimes when we talk about biomolecules, we also take lipid along with it, because during the chemical analysis, when they are treated with trichloroacetic acid and when they are separated, filtered, or when they are strained, this lipid also comes along with these macromolecules. So even though technically speaking, these lipids are micromolecules. Okay, so that's about the macromolecules. Now, as we have already seen, while we discuss about these macromolecules, we have seen that these are usually the polymers. For example, protein. Protein is a polymer of amino acid. So protein is composed of number of amino acids. right? And when we talk about polysaccharides, polysaccharide is also a polymer made up of number of monomers, monomer of sugar. Likewise, nucleic acid. Nucleic acid is again a polymer of nucleotide. Okay, so these number of nucleotides combine, they join together to form a series, or you can say a strand of nucleic acid. Okay, so let us look at some of the bonds, nature of bond linking monomers in a polymer. So bond linking monomer in polymer. So let's first talk about peptide bond. So peptide bonds are the bond formed between two amino acids. So this is one amino acid and this is second amino acid. So in the first amino acid we have amino group and carboxyl group, and again here amino group and carboxyl group here. So there is a bond formation between the carboxyl group of the first amino acid and amino group of the second amino acid. Okay, so during the bond formation, water is released. Okay, so HOH and H, water is released, so thus forming a peptide bond. And if many such amino acids are bonded by peptide bond, it forms a polypeptide. Now next is glycosidic bond. So this glycosidic bond is found in polysaccharides, and we know that polysaccharides are the polymer of sugar monomer. Okay, and that is monosaccharides. So this glycosidic bond, let us just look at, okay, so we'll just look at the symbol actually, sugar, that is disaccharide here, sucrose. Okay, so sucrose is formed by glucose and fructose. Sucrose is formed by glucose and fructose here. Now, this glucose, the first carbon, okay, here it carries the aldehyde group, and the second carbon of the fructose carries the ketone group here. So while forming the bond, okay, they release water and then they form a bond called as glycosidic bond. 1,2 glycosidic bond. 1,2 glycosidic bond in sucrose. Okay, so in this sucrose, you don't find free aldehyde and free ketone because it is lost during the bond formation, glycosidic bond. So such a sugar, sucrose, becomes a non-reducing sugar. It's a non-reducing sugar. Okay, now another example, let's see maltose. So this glucose and glucose, they both combine. Okay, and they are bonded by glycosidic bond to form maltose. So here this glucose at carbon number 1 and next glucose at carbon number 4, they combine, or you can say they are bonded by 1,4 glycosidic bond, and this bond formation is accompanied by the release of water. Okay, so this is a maltose. Okay, having 2 sugar, okay, having 2 glucose bonded by glycosidic bond, 1,4. Okay, carbon number 1 and carbon number 4, there is a bond called as glycosidic bond. So in this sugar, okay, let us say the first sugar, the aldehyde group present on the carbon number 1, it is lost. There is no free CHO. There is no free CHO. And here in the carbon number sugar 2, second sugar, the first carbon, okay, the first position, we have CHO. Okay, free CHO here. So this end is called reducing end, while this end is called as non-reducing end. Now let's look at the bond in nucleic acid. So in the case of nucleic acid, as we have discussed already, it's made up of nucleotide. So nucleotide has sugar here, ribose sugar. Okay, this is a ribose sugar, pentose sugar, this is ribose sugar, and on the carbon number 1, it is bonded with the base. And carbon number 5, 1, 2, 3, 4, 5. At carbon number 5, there is a phosphate group. Same thing you find here. This is a ribose sugar, carbon number 1, 2, 3, 4, 5. On the carbon number 5, there is a phosphate group, and carbon number 1, there is a base. Now, these 2 nucleotide has to be bound. Okay, and it is possible again by the release of water, and the bond formation is between carbon number 1 to carbon number 3, having OH with that of carbon number 5 with that of carbon number 5 bonded with phosphate group. Okay, so 3' OH. Okay, that is the third position. Third position of carbon. 3' OH of first nucleotide and 5' P of the next nucleotide. Okay, so they lose water and thus forms a chain, or you can say thus form a, let us say, dinucleotide. Okay, and such a bond is called as phosphodiester bond. So there's a phosphate group and then there's the ester group here, so phosphodiester bond. So phosphodiester bond is found in nucleic acid, that is in both DNA and RNA. So each nucleotide is bonded to each other by phosphodiester bond. So that's all about the bond formation in macromolecules. Okay, the bonds like peptide bond, Glycosidic bond, and phosphodiester bond. Thank you.